Hey, I'm Stanley Norris, current medical physics registrar at Monash Health, and I'm going to talk to you about radiobiology. This is part 1 of a 3-lecture series. In this lecture, we're going to talk about cellular responses, DNA repair and cell survival, as well as acute and late biological responses, tissue reactions and threshold doses, and a bit on stochastic effects. This talk is heavily based on the Essential Physics of Medical Imaging, 4th edition by Bushberg. These images were shown as part of an exhibition at Melbourne St. Paul's Cathedral in March 2024, which was called 7 Photographs That Shaped the Future. They claim that it could be said that history breaks down into images because they communicate with such immediacy, transcending language and holding stories within them. The moment in which William Röntgen discovered X-rays in 1895 marked a real turning point. This image forms the basis of our work, whether you are a radiographer, radiologist, nuclear medicine physician, medical physicist, and so on. Now, what are the 3 main interactions of photons at diagnostic X-ray energies? Really, radiologists and especially medical physicists should know this. And at which range of energies is each interaction dominant? Well, for energies up to about 100 kiloelectron volts, the photoelectric effect is the dominant interaction. And at energies between 100 kiloelectron volts and 1 megaelectron volt, the Compton scattering interaction dominates. It's important to note that the cross-sections of these interactions, which is related to the probability of an interaction happening, depends on the atomic number of the material that the radiation is traversing through. So this holds true in water. So, why do we need radiobiology? Well, it's not enough to just understand how a medical image is formed through physics. We also need to understand what effect radiation has on biological systems, because the consequences are very serious. Now, going from left to right with these images, you can see a radium girl who suffered from radium-induced sarcoma of the chin. These workers used to lick their paintbrush before painting glowing watch dials. The central image shows the devastation around Urakami Cathedral in Nagasaki in 1945. The effects of radiation took years to be observed in some victims, but many died immediately. And on the right is a technician's hand, mutilated with dermatitis, after working with X-ray apparatus at the Royal London Hospital. These kinds of events provide data that has been used to develop the radiation risk models that we use today. The biological response to radiation will depend on many factors. In terms of radiation, it will depend on the absorbed dose, the dose rate, and the type and energy of radiation. But it will also depend on the cell type and properties and the condition at the time of irradiation. Now, this radiation will result in damage at the molecular or cellular level, but may or may not result in clinically detectable adverse effects. In terms of the way that radiation deposits its energy, we show a simplified picture. So the first step is that incident photons interact in tissue to produce energetic electrons. Each energetic electron produces thousands of lower energy electrons. And these low energy electrons can cause excitations and ionizations in important biomolecules such as DNA. The complex cluster damage, in which groups of several damaged nucleotides occur within 1 or 2 helical turns of DNA, is a hallmark of radiation-induced DNA damage. You can see here these spurs and blobs, which correspond to energy depositions along shorter or longer tracks and slightly different energies. though 95% of energy deposition events from X-rays and gamma rays occur in these spurs. Although these patterns produced by radiation are different from events produced by other processes like chemotherapy, the functional changes to molecules, cells, tissues, and organs cannot be distinguished from damage produced by other sources. Fortunately for us, repair of damage occurs at a molecular, cellular, and tissue levels. A complex series of enzymes and cofactors repair most DNA damage within hours, and damaged cells are often replaced within days following irradiation. So, clinical manifestation of radiation-induced damage may appear after minutes or years. There are two main of bioeffect radiation interaction types, direct and indirect. The direct effect is where the DNA, RNA, or protein becomes ionized or excited by an ionizing particle passing through or near it. In the indirect interactions, it's a result of radiation interactions within the medium that create mobile, chemically reactive species that in turn interact with nearby macromolecules, which may result in the formation of reactive species, which are highly reactive. So, for example, combining to form hydrogen peroxide, that is highly toxic to cells. And most indirect effects are due to interactions of hydroxyl radicals with biologically important molecules. And it's important to note that in medical imaging, 2/3 of total damage is caused by indirect effects. You can see here the indirect and direct modes of action. Now, in the indirect mode of action, the radiation causes ionization and excitation, and the formation of free radicals and hydrogen peroxide. Now, the damaging effect of free radicals is enhanced by the presence of oxygen because it stabilizes these free radicals and reduces the probability of free radical recombination into non-toxic chemicals such as water or molecular hydrogen. Further, the presence of oxygen causes an inhibition of repair because the oxygen binds to the biomolecule and sets damage in place. As you can see, the enzymatic repair systems in the cell can return it to its original state. If a break occurs in a single strand of DNA, the site of damage is identified and the break is repaired. Or if the damage is too severe or repair is compromised, the DNA can suffer mutations and the cell may die. You can see here the timescales also of these different processes. So LET, or linear energy transfer, is just a measure of how densely ionizing a radiation beam is. Shown here in this diagram, schematically represented, low LET radiation is schematically represented by many rays to cause 1 gray of absorbed dose. However, radiation with a high LET is schematically represented by less ionizing radiation for the same 1 gray of absorbed dose. Now, we know that the biological effect of radiation depends on dose rate, environmental conditions at time of radiation, the radiosensitivity of a biological system. But the biological effect also depends on the spatial distribution of energy deposition at the molecular level. RBE stands for relative biological effectiveness, and it's a measure of the effectiveness of a given radiation on the biological system relative to 250 kiloelectron volt X-rays. Therefore, if a radiation has an RBE of 2, that means it needs half as much dose as 250 keV X-rays for the same bioeffect. It's important to note that RBE depends on the endpoint measured. It could be chromosome aberrations, cataract formation, acute lethality of test animals, etc. And you can see on this plot the RBE of radiations with different linear energy transfers. with in increasing LET, cobalt-60 gamma rays, diagnostic X-rays, 1 kV electrons, and alpha particles having highest RBE. Radiation-sensitive targets are located in the nucleus of the cell. So DNA damage is the primary cause of cell death. An energy deposition from radiation induces chemical changes in molecules that may undergo different structural changes. So there are many copies of various enzymes within the cell, and the loss of one copy would not significantly impact the cell's function or survival. If a key molecule for which the cell has no replacement, such as DNA, is damaged or destroyed, this may result in cell death. But cell death doesn't mean physical destruction, but rather loss of mitotic capacity or the premature activation of apoptotic pathways. Rupture of hydrogen bonds that link DNA base pairs may lead to irreversible changes in the secondary and tertiary structures of the molecule. Shown here are some of the important types of DNA damage that can occur. Molecular breakage may involve the sugar phosphate polymers that comprise the backbone of the helical DNA strands, which can occur as single strand and double strand breaks, base loss, base changes, or crosslinks between DNA strands or between DNA and proteins. Single strand breaks and double strand breaks are the most important ones in this context. A single strand break can rejoin provided the broken strands are not separated or oxygen doesn't prevent repair. A double strand break can occur if two single strand breaks are juxtaposed or when a single high LET particle causes a break in both strands. These breaks are highly genotoxic and can result in chromosomal aberrations. Genomic instability from persistent or badly repaired double strand breaks can lead to cancer through the activation of oncogenes, inactivation of tumor suppressor genes, or loss of heterozygosity. DNA damage is actually quite common. Our cells experience thousands of DNA lesions each day. Fortunately, the mutation rate is low because the cell has coping mechanisms. The cellular responses are activation of the G1S checkpoint, which arrests cell cycle progression to allow for repair, and apoptosis, which is programmed cell death, that eliminates damaged genetic material through cell death without releasing inflammatory substances in the surrounding area. Necrosis is an explosive form of cell death that does release material in the surrounding area, which is highly inflammatory. And you can see here a diagram of sources of radiation exposure in the United States. As you can see below, there are many repair mechanisms. This repair occurs rapidly, with about 90% of single-strand breaks repaired within an hour. Non-homologous end joining is the fastest and most common repair for double-strand breaks in humans. It's not perfect and often results in small errors at the repair site. What you can see here from this diagram is in fact the effect of repair. The glowing parts of the cells in the central image correspond to those double-strand breaks. And you can see that 24 hours later, almost all the damage has been repaired. This is a technique known as gamma H2AX fluorescence. Cells grown in culture that are lethally irradiated may fail to show evidence of changes for long periods. However, reproductive failure will eventually occur. The most direct way of evaluating a single cell's ability to proliferate is to wait until cell divisions have occurred to form a visible colony, as you can see in this image. Counting the number of colonies that arise from a known number of cells that are irradiated in vitro allows us to estimate the fraction of surviving cells. With this, we can create cell survival curves, which essentially result from repeating this process for a range of doses that provides a way of measuring radiosensitivity. The survival curves produced from the assays mentioned in the previous slide demonstrate this concept of sublethal damage. When the radiation dose is split into 2 or more fractions, or the dose rate is decreased, cell survival increases. You can clearly see these dose rate effects in the following plot, where a higher dose rate for the same absorbed dose will result in a lower fraction of surviving cells, or higher cell kill. So, it's the same absorbed dose but different dose rates do not give the same biological effect. The alpha and beta parameters can be determined for specific cells and used to predict response. This is key to radiotherapy. We can see here how the principle of relative biological effect, or RBE, in the context of cell survival curves works. Clearly, the radiation types with higher LETs, or linear energy transfer, will kill more cells for the same absorbed dose. So, a higher LET will effectively have a higher RBE. Here are some diagrams which compare low LET and high LET radiations and different OERs. OERs corresponds to oxygen enhancement ratio. Now we can infer a couple of things from these cell survival curves. First, which is that the presence of oxygen increases cell kill for a given dose. If we read off here the 2 gray, the presence of oxygen results in a lower fraction of cell survival, or higher cell kill. The other thing that we can infer from these plots is that higher LET radiation types are less affected by the presence of oxygen. So here again, for the 2 gray, there's a smaller gap between the hypoxic and oxygen curves. Now, a lot of the factors that we have seen so far are known as conditional factors, which exist during or before the irradiation. There are also inherent factors which are characteristics of the cells themselves. In 1906, inherent factors were discovered by performing experiments on rodent germ cells at different stages of spermatogenesis. It was found that inherent factors like mitotic rate and cell differentiation determine radiosensitivity, and you can see in this diagram from most sensitive to least sensitive parts of the body. The stage of cells in the reproductive cycle at the time of irradiation is also a major factor in radiosensitivity. In general, cells are most sensitive during mitosis and the gap between the S and G2 phase. During mitosis, this is due to condensed chromatin, which is more susceptible to radiation-induced damage. Cells in this phase lack robust DNA repair mechanisms also. In the gap between the S and G2 phase, the cell is preparing for mitosis, and DNA is already replicated, making the chromatin structure more exposed. There is also limited repair capacity during this phase compared with others. Now, as we go in decreasing order of sensitivities, cells are then less sensitive during the DNA synthesis preparatory period, or G1. Radiosensitivity in this phase depends on the cell's progression within G1, where early G1 is more resistant and later G1 is more sensitive. And this is because chromatin becomes more structured and compact in later phases, which increases the risk of double-strand breaks, and the increased metabolic activity makes DNA more vulnerable to oxidative stress. Finally, cells are least sensitive during the late DNA synthesis, or S phase. This phase is the least sensitive as DNA is being actively replicated and repair mechanisms like homologous recombination are more active, offering protection against radiation damage. Further, the presence of redundant copies of DNA during replication provides a buffer against radiation-induced double-strand breaks. Cell cycle checkpoints are regulatory mechanisms that ensure the proper progression of the cell cycle. They monitor the cell's internal and external environment to verify that critical processes such as DNA replication and chromosome segregation occur accurately. If errors or damage are detected, these checkpoints delay or halt the cell cycle to allow for the repair, or if the damage is irreparable, trigger programmed cell death. So, this G0 phase is a specialized resting or quiescent phase of the cell cycle, where cells are not actively dividing. Entry into or exit from and regulation of the G0 phase are influenced by signals that can either inhibit or stimulate or even maintain the quiescent state, depending on the needs of the organism and the environment. These processes allow cells to dynamically respond to environmental and internal signals, balancing proliferation, differentiation, and repair with energy conservation and genomic stability. So, what do these effects look like in the clinic? So we're now going to look at the tissue effects due to radiation-induced DNA damage. The response of organs will be different as we're looking at morphological and functional changes in tissues rather than changes in cell survival. We'll see that the higher the dose, the faster the physiologic effects manifest themselves, and that there are practical thresholds below which no effects are usually observed. In terms of the organ system, the type of the response and its severity will depend on the dose, the volume of tissue irradiated, its radiosensitivity parameters, and the regenerative capacity of that organ. So upon a radiation dose, if the dose is too high, the organ system may undergo tissue necrosis. Otherwise, healing may happen. In one case, you may have repair, in which damaged cells are replaced by a different cell type. This is fibrosis, and in this case, the organ is not returned to its pre-irradiated state. Otherwise, regeneration can occur, and this is a replacement of damaged cells by the same cell type, so the organ can return to its pre-irradiated state. Now, you can see here some of the radiation-induced effects on the skin, which are pretty grim. Radiation-induced skin damage is the most commonly encountered tissue reaction following high-dose, image-guided interventional procedures. These injuries can be severe, debilitating, and in some cases, as in the bottom right, they can cause chronic necrosis and require surgery. There are often no immediate symptoms from skin doses, and symptoms may take weeks or months to appear. Skin damage is a consequence of oxidative stress, which results in a cascade of inflammatory responses, impairment of stem cells, endothelial cell changes, and epidermal cell death. Skin reactions have a threshold of about 1 to 2 gray, below which no effects are usually seen. This table indicates what tissue reactions can be expected following an acute skin dose. For example, temporary epilation can occur about 2 to 8 weeks after an exposure of 2 to 5 gray. After a large dose, like 20 gray in a single hit, intense erythema, followed by acute dermatitis and moist desquamation, can occur with permanent hair loss and damage to vascular structures. With failure to heal, ulceration can occur, and in very high doses, so can necrosis. Moist desquamation is a clear predictor of late delayed injuries. It's important to note, though, that a patient will have individual radiosensitivity that depends on factors like the exposure area, anatomical location, fractionation, overall health, medications, and so on. It is not exactly deterministic. In general, the gonads are very radiosensitive. The primary effects on male reproductive system are reduced fertility and temporary and permanent sterility. Temporary sterility can occur after a dose of about half a gray and permanent sterility after about 6 gray. The duration of sterility is dose dependent, with the recovery taking years. However, there will be a window of fertility before the onset of sterility, as long as mature sperms are available. Now, you can see in this plot here the risk of different effects during the gestation period. For example, the most likely effect of a radiation dose during pre-implantation is prenatal death, while the most likely effect to a fetus is growth and mental retardation. We can also see how this applies to mice, although their gestation is much faster. The lens of the eye contains a population of radiosensitive cells that can be damaged or destroyed by radiation. But since there's no removal system for these damaged cells, they can accumulate to the point where they cause vision-impairing cataracts, as you can see in the image. These cataracts are caused by abnormal differentiation of the damaged epithelial cells that begin as small opacities in the anterior subscapular region and migrate posteriorly. The degree of opacity and the probability of its occurrence increases with dose, and the latency period is inversely related to dose. Studies of atomic bomb survivors, workers in the Chernobyl accident site, and radiation technologists in the USA indicate that the dose limit to the eye is about 20 millisieverts per year. There is a general agreement that acute doses below 100 milligray will not result in functional impairment of tissues or organ systems. As physicists, or people working in healthcare, we should try to have this number in our head. So the consensus here is that 100 milligray is the approximate dose threshold for tissue effects. As you probably already know, it's well established that radiation can increase the risk of cancer years after exposure. While moderate doses of radiation cause well-documented effects, one cannot detect significantly increased effects at doses typically encountered in diagnostic imaging. Here we provide a very, very simplified picture of carcinogenesis, which is that cells in a tumour are believed to descend from a common ancestral cell that at some point loses its control over normal reproduction. The malignant transformation of such a cell can occur through the accumulation of mutations in specific genes. Cancer is a process in which the initiation of damage within a single cell is followed by a sequence of events that permit the cell to successfully proliferate. For the purpose of setting radiation protection standards, it is assumed that there is no threshold dose for cancer induction. So, during initiation, a somatic mutational event occurs that is misrepaired. During the promotion stage, the cell is stimulated to divide. A promoter is an agent which alone does not create cancer, but once an initiating event has occurred, it promotes or stimulates the cell that contains damage. Now, the final stage is progression, during which the transformed cell produces a number of phenotypic clones. Eventually, one phenotype acquires the selective advantage of evading the host's defense mechanisms, thus allowing the development of a tumor and possibly even metastatic cancer. Radiation may also enhance progression by immunosuppression resulting from damage to lymphocytes and macrophages. Thank you for listening, and another gentle reminder that this lecture is heavily based on the essential physics of medical imaging by Bushberg, and all visualizations, diagrams, and graphs, unless otherwise stated, were taken from this book. Thank you.